Okay. Title of my talk is Tracking Vector Safety in Gene Therapy. In other words, also looking what is happening with the vector, especially where does it integrate in the host cell genome. A short outline, we'll briefly go over the integration site analysis or the tracking itself. Simply give you a brief overview of the clinical gene therapy studies with regards to genotoxicity or also safety in the Gammaretroviral vectors, lentiviral vectors, very brief with adeno-associated viral vectors, and then also the challenges in tracking the clonality or also the opportunities we have nowadays in the next generation sequencing area. So the idea behind the clonality analysis or integration site analysis arose from the 80s of the last century, where the idea was, in the end, the first gene therapy studies were gene marking studies, really being able to mark any cell of interest, like here shown for a blood stem cell. If genetic information from outside is given, like a retroviral sequence or a retroviral vector, this vector with an integrase function will integrate into the host genome, and thereby, here it's shown in red, will mark each individual cell according to the position where the vector landed. So cell 1 will differ from a transduced cell 2 simply by the vector location or by the integration site, which in the end then brings us really to gene marking studies also. If a progenitor cell, for example, blood progenitor cell or stem cell is transduced, for example, 3 months later after reinfusion into a preclinical model or in a patient, one can sort different lineages and look whether a T cell shares the same integration site like a granular site, where we then can retrospectively say, initially, okay, we have transduced a very early progenitor cell. The challenge, also when I stepped into this area, it was in the year 1995, was that there was no real method available. There were also another challenge that the human genome was not sequenced, also the mouse genome was not sequenced. But primarily it was a technical challenge. Available methods were the groundbreaking ligation-mediated PCR and inverse PCR, but these both methods were not sensitive enough. So we developed a linear amplification-mediated PCR. We had shown the principle. The idea is here is the vector integrated in the genome in an unknown region of the genome, simply to identify this vector genome fusion sequence. And the idea of the LAM-PCR was simply like old primer extension preamplification protocols for whole genome, simply to enrich for these fusion sequences. So the method itself, one is using biotinylated primer. These are extended in form of a linear PCR into the genome. Then we make an enrichment via streptavidin coupled paramagnetic beads, so that our target DNA is selected. Afterwards, we build up the complementary DNA strand. Then we perform restriction digests, normally multiple restriction digests with multiple enzymes, because we do not know where the next restriction site is located on these fragments. Then we perform a linker ligation. We simply add a known oligolinker cassette to the unknown genomic DNA, so that in the end we can do a conventional PCR, because we know the vector sequence here, this part. We do not know the genomic part, but then we have again a known sequence at a 5' end. This can then be amplified exponentially and sequenced. Here is exemplarily shown The first two lines show you an oligoclonal to polyclonal situation. The right line is a monoclonal situation. So each band corresponds to one specific integration site. In the end, it's an RFLP, restriction fragment length polymorphism. We have done this then quite early in clinical studies. The first clinical studies we did this was an ADA-SCID gene therapy study, one of the first conducted in Los Angeles by Donald Cohn, where we analyzed 2 patients over a time frame of 9 years. And as you already see here, you see individual bands, some more, but it looks very monoclonal, so mono to oligoclonal. So these samples were taken from peripheral blood and also from sorted T cells and also monocytes. And what we saw here is that the long-term lymphopoiesis in these patients, the trial was not really effective. There were also no side effects. So this I would like to pronounce here. That the lymphopoiesis, long-term lymphopoiesis was driven mainly by 1-2 single clones, and this for both patients. So we had here clear clonal dominance. I think it's very important because a lot of people say clonal dominance means the first step of malignancy. This must not be the case. This is not stringently linked. For sure, there were other examples where with severe adverse events which were not foreseen. An example from the SCID1 gene therapy trial that was conducted in Paris. Here's the results also of the integration site analysis for one patient and another patient. And what you see here in the beginning, 6 months, 13 months after treatment, a very polyclonal situation, which then, starting with 24 months, got more and more, became more and more monoclonal situation. And then also 2.5 years after treatment, tumor was determined. It was in the TALL, acute lymphoid leukemia, in this patient, also in the other patients. In both patients, it was obvious when we then sequenced this dominant clone or leukemic integration site, we found that the vector was integrated near the proto-oncogene LMO2. So here an example for really drastic case where leukemia occurred in treated patients. So in the end, this also then triggered really all the safety discussions in this regard and genotoxicity, because before, we just had a talk before with the animal models. In none of the animal models till the year 2002, any malignancies that were related to the treatment, to the vector, have been observed. Here in these early studies, Gammaretroviral vectors were used, which was also used in a Viscon-Aldrich syndrome gene therapy study in Hanover, where also the outcome was very dramatic after 1.5 and 5 years for individual patients. 7 out of 9 patients acquired a tumor, 6 ALL, and 3 patients acute myeloid leukemia. And 2 of these AMLs were secondary after the onset of the ALL, and then after treatment, AMLs occurred in these 2 patients. What is common to all of these patients when we looked into the peripheral blood over time, where the vector was located, which clones were existing. You see here each color represents one patient, and really in this suspected genes or known proto-oncogenes that have played a role in previous ancestral oncogenesis, like MDS1, EV1, and LMO2. We've seen a lot of integration sites that were clustered, a lot of clones that had the vector located in these gene areas. So this study was also conducted with the Gammaretroviral vector. So it was clear there was also a septic CGD trial where 2 patients or 3 patients in Frankfurt and Zurich acquired MBS, myeloproliferative syndrome. It was clear that insertional hematogenesis really has an active role or increase may accompany the gene therapy with an increased risk simply, which is for sure given the kind of the vector that was used in these studies was a conventional Gammaretroviral vector which was Full-LTR driven. This means the LTRs, the long terminal repeats that include the enhancers and regulatory elements of a natural virus were present 2 times here on the 5' side and the 3' side. And as we now know today, the risk of oncogene activation is simply drastically increased. But here I also would like to note that also Gammaretroviral vectors have been used in ADA-SCID. Meanwhile, I think there are more than 50 patients worldwide, and there was so far not any case of insertional mutagenesis event. So that one clearly has to see, which is not fully filtered out here today, whether the disease background plays a role, whether the transgene itself plays a role, whether it triggers, whether it gives the transduced cells, the gene-corrected cells, a substantial proliferative advantage. So these things are still ongoing, also in a variety of research labs. Anyway, it was then clear that this kind of vector, if using retroviral vectors, cannot be used in the long run. So then the self-inactivating LTR lentiviral vectors came onto the stage. That was one main feature for sure is that the LTRs, the regulatory elements of the LTRs were deleted, and that internal promoter was used, ideally nowadays a tissue-specific promoter, simply reducing the risk that this new genetic information may trigger any unwanted cellular gene expression is drastically minimized. And this could also hold true then in the studies, in the clinical studies that have been conducted, like in the adrenoleukodystrophy, but also in metachromatic leukodystrophy or Viscott-Aldrich syndrome, or also for beta thalassemia, where one then could really say, okay, gene therapy that works. This was in 2013. This editorial from Indoverma. Where one could really say, okay, also again here comparison, the current generation lentiviral vector, very self-inactivating LTR composition has an increased, represents an increased safety feature. Another feature for sure is also that the natural integration characteristics of lentiviruses and also the derived vectors are different from Gammaretroviral vectors, because the Gammaretroviral vectors tend to integrate near gene promoter regions, whereas the lentiviral vectors are more uniform distributed. Also, though they have a preference for gene coding regions, also because these genes are simply expressed, they are more distributed over the complete gene and do not cluster at the promoter regions. Is then also, if you look for the Viscott-Aldrich syndrome, and if you make a direct comparison between the Gammaretroviral trial and also the lentiviral trial that was conducted in Milan, we see that the Gammaretrovirus, if we analyze the peripheral blood, we see increased or very drastic hotspot in the MDS1 EV1, or nowadays it's called MECOM gene region. Whereas with the lentiviral vector, there's on the one hand not such a hotspot, and this one EV1 is a proto-oncogene, and there's a more safe distribution of these vectors also with regards to the genes or to proto-oncogenes. Simply only one slide for AAV, which is where we could recently show about 2 years ago in the Gammaretroviral, so the first gene therapy medicinal drug, that also here integrations may occur, though this vector does not have an integrase function, but it's very likely simply given by the natural occurrence of double-strand breaks in our cells, in each of our cells, and then the DNA repair, mainly by non-homologous end joining, so that in a low frequency, around 1%, also integrated vector, AAV vector, or also plasmid, this we have also analyzed in vitro, may simply occur, may simply be captured into the host cell genome. But here, I would like to pronounce, we have checked this and we found really a random distribution, a uniform distribution, and we do not find any integration in one of the genes or gene regions that have been also reported recently, just a few weeks ago, where Nord et al. Found wild-type AAV2 in human hepatocellular carcinomas, and from there suggests that AAV2 may play a role in the formation of these carcinomas. So all genes that are listed there, we do not find them. We also do not find them in another clinical trial which is directed to treat porphyria, acute intermittent porphyria, where we also have liver biopsies from the patients and also from the associated non-human primate model. And also there we do not find these HCC, AAV suggested or proposed genes. For this I would like to refer you to Irene's talk tomorrow in session 1B for viral vectors, and also to the poster session from Raffaele, where he will present also some more insights into AAV concatenarization and rearrangements. So nowadays, as I mentioned, we are in the next generation sequencing area, which gives a lot of new opportunities, for sure also challenges, especially if we're talking about millions of raw sequence reads, and really how to handle them also for the data analysis. Here simply some features what we have done, what we have improved. So the original protocol is simply the LAM, you see it here for sample 1, for sample 2, for sample 3. Normally, in former times with Sanger sequencing, we simply sequenced one sample. Then with the arising from 454 pyro sequencing, where we reached up to 800,000 raw reads and not only 96, we started with multiplexing and we added a barcode to these samples here at the last step, so that we then could mix sample 1, 2, 3, sequence them in one sequencing run, and then afterwards sort against the barcode and analyze for each sample integration sites separately. What we have now changed already in the context of the 454, but now we're doing also MiSeq, sometimes also high-seq Illumina sequencing analysis, and with the MiSeq already there we reach about 15 million of reads in one single run. We added another barcode which differs from the first barcode, but this other barcode, which I think is very important, or what we see also from collision rate or contamination rate, we add this first barcode already at the ligation step, where no exponential PCR is ongoing. And you see here some examples, and also I only want to focus on this part. If you look here for the portion, the ratio between blue are the integration sites that we find, and here in brown, light brown, red are the collisions, so that we find the exact integration site in sample 1 from patient 1, in another sample from another patient where simply the likelihood having the same integration site, the same clone is very low, you see that here we can drastically reduce this kind of collision or cross-contamination so that we really can be sure with a high probability that the integration sites we retrieve from one sample really belongs to this sample. Here is, if you compare it also with the MiSeq is more dramatic because of the high reads. If you are using only a single barcode, even the brown is higher than the blue. So the collision or collidide integration sites are higher than the real integration sites we find. Other example for sure, if one switches from the 454 to the MiSeq, with the MiSeq one has simply the opportunity to reach, to go to saturate the sequencing, which then allows us also, you see a direct comparison here, the 454 MiSeq, 454 MiSeq, also from patient sample, but the 10 strongest clone here underlined in blue are really the same, but the total number is quite different, so we find up to 20-fold more integration sites in our peripheral blood sample compared to the 454 with lower sequencing reads. Simply to give you also an outreach, so far I have shown you the LAM-PCR. There are a variety of PCR-based methods out there, but to a certain degree we also wanted, also given by our experience with the cancer genomics that is ongoing in Heidelberg, in line with all exon studies, where simply all exons are captured and then directly sequenced. We are also pursuing the target enrichment sequencing of vector sequences. And this is simply the idea, so you would to avoid as much as possible any PCR, so that we also have the possibility to get a very accurate quantitative output. So how strong is the respective clone where the integration site belongs to. So the idea is simply you take your DNA of interest, the genomic DNA of interest, you share the DNA, you add the sequencing specific adapters, and then you give complementary probes to the vector sequences so that you really retract or extract the vector specific sequences, and then you can directly sequence them. Other opportunity would be whole genome sequencing, but this is already nowadays still too expensive in that regard. So we have done this with very promising results. I think we also made for sure some control runs. Only to show it to you here to you. So if this is from a simple monoclonal transduced cell line. So if you do this target enrichment, here is shown the 5' integration site. This was from a lentiviral vector. Here it's the 3' integration sites. We have left out the complete vector sequence so that you really can nicely see also the 5 base pair overlap that is generated by the lentiviral integrase. The main idea, the main hope we have here is also what I mentioned with the quantitation of individual clonal contributions, because in an idealized world, if we one could also hybridize or target capture only the LTRs of a vector, here shown in blue. So then one could go simply by the sequencing depth or the sequence count, how often is one specific integration site sequenced, and one can correlate it directly to the complete number of the vector. And in addition, if one would use a FID probe, a subgenomic probe, for example, for normal housekeeping gene sequence or exon of such a gene, one should also be able to make an absolute quantification of the vector presence in the analyzed sample. Only one result here, where we compare this, so in the end avoiding a real qPCR that has to be done also on these samples. If you correlate the qPCR with this target enrichment sequencing, where we also have, together with vectors enriched for subgenomic region, you see a very high, a very good correlation with an R coefficient of 0.93, so that really this kind of quantification of vector genome ratio is working very well with this target enrichment approach. So here simply the idea of this target enrichment sequencing is, on the one hand, it's non-PCR, it should be more accurate from the quantitative aspect. It should be, which I think is very important, also should be independent from the sample size, and to some degree also from the sequencing depth if an additional subgenomic region is captured, so because then one can directly correlate the sequence counts with the vector sequences and the vector integration side. It should also, for sure, consequently gives a high QC in this regard, and it should be easily possible to scale it up and to automate. Stefan Wilkening will have a talk tomorrow and will give you more insights into this target enrichment sequencing of vector integrations. And also Karl Petri in the Brüster session tomorrow will show the target enrichment of wild-type AAV sequences. For sure, simply some few words to the bioinformatics. For sure, with the conventional manual analysis, there's no chance. Also with our in-house pipeline we have generated about 5 years ago, there's simply no chance, because if you're really looking for millions and millions of sequences in parallel, so our initially implemented TISA pipeline sometimes takes 2 to 3 months. So for this reason we have implemented a new pipeline also based on the BUAW primer, also known from the omics people, which allows us to analyze LAM-PCR sequences, but also like for the target enrichment, paired and sequence reads. It's the bioinformatical suite is composed simply by different modules. Normally one has the input, we make a pre-processing, in the end we really do 2 different alignments and post-processing annotation. So which for us is very important, that it's very accurate, and that we, for example, here's a typical time frame for 15 million raw sequence reads we retrieve. Normally only 13 minutes of pre-processing time is needed, and we came out with the annotation done within another 30 minutes. What we're currently then implementing are the downstream analysis, so that we not only can correlate which gene is located next to this integration site, but also which microRNA, which perhaps also we call it transcriptional atmosphere, is located there, and some other features. But here also would like to refer to Salva, who has developed together with Raffaele this bioinformatic or computational tool, and who will have the poster also tomorrow. So in the end, to summarize, we have built this up over, I think now 15, almost 20 years, a large clonality and integration site screening assay, starting really with the clonality, now focusing more and more on the safety, also from the SAEs or adverse effects that happened in few clinical trials and few patients therein. But based on this screening assay, we have enlarged over the last years also our field of interest. So we're going also to cancer drug resistance screening, and also for sure immune repertoire studies, and also gene editing of the TCR. Christoph, our director, will have tomorrow a talk, and Friederike will present tomorrow T-cell receptor modification by TALEN and CRISPR, and Tim will show how we try to address the cancer drug resistance screening also tomorrow. For sure, I would like to know that for conflict of interest or to disclose this, I'm co-founder of a company. And here I would like to end. I would like to thank all lab members, and especially also all former members from our group, our sequencing core facility at the DKFZ, and especially all collaborators collaborators that had made all this kind of research possible, and also here Net4CGD and other third-party funding agencies. Okay, thank you.